Next up, we're going to talk to move beyond those larger organelle structures, and we're going to still talk about some of the other organized and complicated membrane-bound vesicles that play an important role in the cellular function. So the ones that we're going to look at next are called microbodies. Microbodies are going to be membrane-bound vesicles that contain specialized enzymes that perform a very specific metabolic function. So a great example of that would be peroxisomes. Peroxisomes are going to be membrane-bound vesicles. They're going to have an enclosed they're going to enclose certain enzymes that are going to be responsible for breaking down hydrogen peroxide into water and carbon dioxide. So they have a very specific function. Another great example of these would be lysosomes. Lysosomes are going to be vesicles that contain specialized enzymes or a specific range of pH that's going to be used to help digest and break down unwanted waste within the cell. So those are going to be our microbodies, and then also there are vacuoles. Vacuoles are kind of interesting. Vacuoles are going to have just a couple of interesting characteristics. They're going to be membranous sacs. They're going to be much larger than vesicles. And their primary role is going to be storage. However, inside of a plant, they're going to have one other purpose. Inside of a plant cell, a plant vacuole is going to be very large and located in the center of the cell. So it's going to be a large central vacuole, and it can take up to 90% of the total volume of the cell. Its primary role is going to be storage of nutrients, pigments, water, waste products, but it's also going to be responsible for developing turgor pressure. Turgor pressure is what allows your plant to stand upright, and it's also the loss of turgor pressure is what allows your celery to become rubbery if you let it sit out on the counter, or allows flowers to wilt after time. You can kind of think of the plant vacuole as like a balloon inside of a box. You stick a balloon inside of a cardboard box, and then you blow it up as big as you can until it pushes against the walls of your cardboard box. Now, your cardboard box might be pretty steady, but it probably can't support your weight. It'll just collapse. If, however, you have a really, really full balloon on the inside pushing against all of the walls simultaneously, that cardboard box is going to hold your weight. It's going to support your weight. If, however, you were to deflate that balloon, you would suddenly find that box collapsing in on itself again. This is how the plant's central vacuole helps to keep the plant upright. When it's full of water, the plant will stand upright. Those cardboard boxes are nice and rigid. Those cell walls full of cellulose are nice and rigid. If, however, you start to lose water, if the plant starts to wilt due to a loss of water, that turgor pressure is going to decrease, and the cell walls are going to just kind of soften. They're just going to kind of weaken and collapse in on themselves. We're not going to turn the plant into soup. Like I said before, those cell walls are still made up of cellulose. They're going to still be pretty rigid, but it will collapse in and sink in a bit. Vacuole can also perform or can also hold toxic substances that are used for protection against herbivores. If you come across a plant that is poisonous, most likely that poison is going to be located inside of the vacuole. This is a fantastic storage space for toxins because they're kept away from everything else inside the cell. It's going to keep the plant cell from accidentally poisoning itself due to those toxins. And so this is going to contain a lot of toxic substances that are found in poisonous plants. It may also perform some of the functions of lysosomes in other eukaryotes. That's it for our endomembrane system and our microbodies and vacuoles.